I'm going to go ahead and start. I'm going to finish chapter 3. I'm going to go back to, a little bit previous to where I finished. And I want to get to at least a good stopping place in chapter 4. Well, there will be some homework posted on Canvas that concerns itself with chapter 3. And I'm going to do some searching on Connect for some more homework that devotes itself to chapter 3. For example, probability calculations and chi-square analyses and things like that. And that's just because I don't directly have anything I can upload to Canvas that concerns itself with that. And so I'm going to hope that somewhere on Connect there are assignments that will deal with some statistics in chapter 3. And so let's see, I got to in chapter 3 right about here. And at the time I got there on Tuesday, I was getting tired and I didn't do a really good job. And so I want to go back. Now, I went through pedigree analysis and I ended up basically going through probs and stats. And there's a definition of probability, which is very simple. And there's predicting the outcomes of crosses using a variety of equations. And one of them is the multinomial equation, which I want to know that it exists. But this book does not carry out any kind of detail about using the multinomial equation. And I have forgotten most of how to use the multinomial equation. But the laws of inheritance can be used to predict the outcome of genetic crosses. And some of this is fairly complicated, and some of this is fairly straightforward. There are some concepts in genetics that we get to in a later chapter in this textbook. And one of the things you might want to know about is genetic by environment interaction. And so a lot of phenotypic traits are explained almost 100% by genetics. And some phenotypic traits are explained almost 100% by the environment in which the animal or the plant is raised in. And there are some traits where there is a significant genetic times environment interaction. And these probability calculations can allow plant and animal breeders some kind of knowledge about how much of a genetic variation concerning the trait in question has been exhausted by generations of selective breeding. And then in the human world, parents might be interested in predicting the traits that their children might have. And this is especially important in the case of families that have a history of genetic diseases. And the probability of an outcome is just the likelihood that it will occur. So it's just number of times an outcome will occur over the total number of possible outcomes. So for a coin flip, probability is an italics P of a head resulting in a single coin flip is 1/2. Because you get 1 in 2 chance of it being a head or a tail. Now, in genetics, if you cross 2 heterozygous pea plants, and they are both heterozygous for purple flowers, and so the probability of you observing, or Paul plants or whatever, the probability of observing the dominant phenotype is 3 out of 4. And the probability of observing the recessive phenotype is, well, it's going to be the remainder. The sum total of all probabilities must equal 1. And so there's 2 ways to calculate this. Probability of the dwarf is 1 minus 75%. which gives you 25%. Or you can go back to the Punnett square and figure out how many ways you can get a dwarf versus how many total phenotypes you might observe. And then it talks about random sampling error. And in genetics, especially, the larger the sample size, the more accurate your predictions can be. And that is just due to random error. And this random error is relatively large for small sample sizes, and very small for large sample sizes. And so they give you the example. If a coin is slipped 10 times, it shouldn't be beyond the scope of your expectations to get a 70-30 ratio. If you flip it 1,000 times, you're much more likely to see that 50-50 ratio. And for example, this is why when, say, for example, polls are taken to predict election results, this is why the number of individuals they sample is usually in the thousands. And that's because it makes it far more accurate of a predictor of the election results. Now, there is something called the product rule. The probability that two or more independent outcomes will occur. And it's calculated by the product of their individual outcomes. And independent outcomes mean the occurrence of one outcome does not affect the probability of another. And so I'm going to apologize for the alleles they have assigned to congenital analgesia. It's a recessive trait. And capital P is a normal allele. Lowercase p is the recessive. And so you have a question. Two heterozygous individuals for congenital analgesia plan to start a family. What is the probability that the couple's first three children will all have congenital analgesia? And this is where I'm going to apologize. The probability for any given individual from the above setup having congenital analgesia is 1 in 4. Probabilities that the first 3 children will suffer from this are 1/4 times 1/4 times 1/4, which apparently gives 1/64th. And if you want to put that in decimal, it comes to about 1.5%. And that's the probability that the first 3 offspring will all have that disease. And you can extend this to multiple genes. And so you have this cross. Individual heterozygous at A, heterozygous at B, homozygous dominant at C, crossed with an individual heterozygous for A, homozygous recessive for B, heterozygous for C. What's the probability they will have an offspring with a genotype? Capital A, little b, capital C, little c. And it's assuming that these are inherited independently. Probability that an offspring will be capital A is 1 in 4. Probability that it will be little b, 1 in 2. Probability that it will be big C, little c, again, 1 in 2. And you just multiply 1/4 times 1/2 times 1/2. And you get about 6.25% of probability of having exactly that genotype. Now, there is another equation. It's called binomial expansion equation. The probability that the unordered outcomes laid out in the problem will occur. And n is the total number of outcomes. X is the number of outcomes in one category. N minus X will be the number of outcomes in the other category. P is the individual probability of outcome X, and Q is the probability of the other outcome. In population genetics, you can denote the alleles P and Q. And P usually means the frequency of the dominant allele, and Q usually means frequency of the recessive allele. And if those are the only two alleles in the population, then they must add to the sum total of all the alleles in the population. And simply the explanation point means a factorial. And so it's just called bang. So 4 bang is the product of 4 times 3 times 2 times 1. And 5 bang will be 5 times 4 times 3 times 2 times 1, and so on and so forth. The exception is that there is a mathematical definition of 0 bang equals 1. And so you have 2 heterozygous brown-eyed individuals have 5 kids. What is the probability that any married that any 2 of the 5 will be blue-eyed? And so you can use the binomial expansion equation. And you can do this via Punnett square. You can do this mathematically. The probability of a blue-eyed individual is 1/4. Probability of a brown-eyed individual is going to be 3/4. And n is going to be the total number of events. In this case, it equals 5. X is going to be the total number of blue-eyed children. In this case, it equals 2, because it's set up that way in the question. And now that you know P and Q and X and N, you just plug those values into that equation and do the math. And it comes out that the probability is 0.26, or about 26% of the time. And there's the multinomial equation, and just know that it exists. Now, you might be asked to do some hypothesis testing. And a very simple way to see if your observed data matches your expected data is chi-square test. And all it does is it examines whether the data that you observe and the data that you expect are statistically different from each other. It doesn't prove anything. It just mentions that, goodness of fit. And so, for example, you look at a dihybrid cross. So you look at the fruit flies, and you have the allele C plus, which is normal, and C, which is curved. And then you have body color. E plus is normal, which is grayish, and E is ebony. Now, in fruit flies, the wild type allele is designated with that plus, and anything without the plus is some kind of mutant. And so here's the general formula of the chi-square test. It's the sum of the observed minus the expected squared divided by the expected number of individuals you expect in every category. And so you do the cross between the true breeding flies, wild type, and mutant, and that gives you the F1 generation, which will be heterozygous for both alleles in question. And then you cross them to make the F2 generation. And these are the raw numbers that are observed. And notice that if you do the math, this is not an exact 9 to 3 to 3 to 1 ratio. That would be very unusual that you would get exactly 9, 3, 3, 1. And so to use the chi-square test, you have to make a null hypothesis. Your null hypothesis for this kind of cross is that the two traits are independent. And therefore, you would expect 9, 3, 3, 1. And if you did the math, that 9 would be 9/16 times 352 for 198. 3/16 and 3/16 would be 66 and 66. And then the double mutant phenotype would be 22. And if that mathematics doesn't come out, you have fractions, you have a decimal, you can either round up or round down. You can drop it, or you can round it up to the next numerical value. Or you can leave it alone. It really doesn't matter. And now you apply the chi-square formula, which is the sum of the observed minus expected in category 1 squared divided by the expected number of category 1, and observed minus expected for category 2 squared divided by the expected number in category 2, and so on and so forth. And there is your observed data minus the expected squared divided by 198. And then your observed data minus the expected squared divided by the expected, observed minus expected squared divided by the expected, and then observed minus expected squared divided by the expected in category 4. And you add all those things up together. And it comes out that your value that you calculate is 1.06. And now you have to interpret that value. Now, here are the chi-square values that you use. Now you have to look at that table and see. Now, the lower this number is that you calculate here for chi-square, the more likely it is that the observed values that you see are due to random chance alone. The bigger this number that you calculate means that it's a very low probability that the observed deviations are due to random chance alone. And in this case, looking at the hybrid cross, what this high chi-square value would indicate is that these genes are not inherited independently. They, in fact, are linked. And typically, you test the results at the 0.5 level, so less than 5%. And if the chi-square you calculate, and in fact, in this example, if it's below 7.8 and some change, that means your null hypothesis is supported. Now you have to determine what they call degrees of freedom. And the degrees of freedom is a measure of categories that are independent. And in this case, the degrees of freedom is going to be 3. Because in dihybrid cross, there are 4 possible categories, and you have to penalize yourself because you did some manipulation of the data. When you did this, that was manipulation of the data. You did one manipulation, so you have to penalize yourself by 1 degree of freedom. And so you calculate the number of degrees of freedom, which in this example was 3. And you test against the 0.5 level. And you're looking for a number that's greater than or less than this 7.815. And if the chi-square value that you calculate is greater than this, that means your null hypothesis is not supported. If it is less than that, that means your null hypothesis is supported. That is to say, if the number that you calculate is less than 7.815, the odds are very good that the observed data deviated from the expected data in a non-significant fashion. That is to say, it was within the realm of random chance, random sampling error acting alone. And so, with 3 degrees of freedom, and you calculate the chi-square value of 1.06, it is approximately at about that level. More importantly, it's well below here. And so that means that the numbers that you actually observed and counted up should be within the realms of random chance. Therefore, it is about 80% probable that random sampling error explains the data that you saw. Therefore, you accept the null hypothesis that genes C and E were indeed independently inherited. I hope to find some chi-square analysis stuff on Connect. Now, this, I hope, is mostly more review. Okay. For sexually reproductive species, there are usually 2 opposite sexes. And so, a variety of different species rely on different ways to determine sexes in the organism in question. And for some species, the sex determination is chromosomally based. And for example, in a mammal, we use the terms chromosomes X and Y. In a lot of birds and some other organisms, we use the chromosomal indications of Z and W. For some organisms, it is the temperature at which the egg is incubated that determines maleness or femaleness. And for some organisms, it's behavioral interactions. And that can be the distance away from the mother that the embryonic organism matured in, or it can be something very different than that. But in mammals, we say they use the XY sex determination system. And the male is XY, or the heterogametic sex, because this allows the male to make two different kinds of sperm, one carrying the X and one carrying the Y. And the female is XX, homogametic. All gametes are going to carry an X chromosome. And then one of the 22 autosomes in both cases. And in this type of sex determination system, it is the male that determines the sex of the offspring. And so we know now, and I don't like the fact that it's a single gene. I learned in a sex determining region Y, and so it's a few genes. But anyway, we now know that it's the presence of the Y chromosome that promotes male development. And the absence of a Y chromosome is going to promote female development. And we know this from a variety of things. But one of the pieces of evidence is because you can have individuals that are XXY and they develop male. And you can have individuals that are X nothing and they develop as female. And it turns out there's a little region on the Y chromosome that is responsible for this decision that is made. Some organisms, they use what is called the X0 determination. The male is X nothing. The female is XX. In the X0 system, it is actually the ratio of X chromosomes to autosomes that is the actual determination. And so if it's one X to two autosomes, it's male. So if it's a 1/2 ratio, it's male. If it's a 2-to-2 ratio, it's female. And then you have ZW. In this case, the male is ZZ and the female is ZW. And in this case, it's the female that determines the sex of the zygote. And in some animals, there is a haplodiploid sex determination system. And it's found, for example, in bees and some wasp ants. In a honeybee, the male is haploid. And in that bee, the females are diploid. And so the male results from an unfertilized egg, and the female develops from a fertilized egg. Just to make life more difficult. And then in some things, it's temperature dependent. For example, in an alligator, eggs that are incubated at temperatures equal to or greater than 34 degrees centigrade become male. If they're incubated at below 30, they become female. If they're incubated somewhere between 30 and 34 centigrade, flip a coin. And in this species, in alligators, male and female of the species have the exact same chromosome makeup. And then there are behavioral determinations. For example, in the genus Amphiprion, which include the clownfish, all the clownfish are born male. They're proto-androus hermaphrodites, androgyny first, and they will switch from male to female. And so female comes later. And so in the clownfish, they have a symbiotic relationship with sea anemones, and there's a population of fish that lives within each anemone. One is an adult female, one is an adult male, and lots of juveniles. If the adult female dies, the adult male switches sex and becomes female. And one of the juveniles becomes an adult male. And so, again, the male and the female clownfish have the exact same chromosome makeup. I like that because it ruins Finding Nemo for all of my students. And then you have plants. In plants, the typical plant produces both male and female gametes on the same individual plant. Some plants are dioecious. That is to say, each plant is either going to be a male or a female. And in some plants, we'll fall back on the XY system. And we'll say that the male is XY and the females are XX. But for other plants, you can look at the karyotype all you want. And there is absolutely no visible way to recognize different sex chromosomes. And so how gender is determined in these plants, we don't know. And so anyway, there's pictures of random plants that might be male or female. Now, there's some kind of hand waving. It's easy to explain why you have two copies of every chromosome. That means if you assume that there's one gene on each chromosome, that means you have two copies of each gene. And so that means the dosage of the genes that you get is equal. And it's easy to determine that you need the same gene dosage for normal development. But for genes on the X and Y chromosome, males and females have different numbers of X and Y chromosomes. And so they have different numbers of genes that might be found on those chromosomes. And there's got to be some kind of explanation why this different dosage at these genes on these chromosomes doesn't cause problems. And there is at least one observed phenomenon that you find in mammals. It's called X chromosome inactivation. If you have more than one X chromosome, all of them except for one will be inactivated, and the genes on it will not be expressed. And that's pretty normal in male mammals. In female mammals, that causes some peculiarities. And so here is the concept of dosage compensation. You have to do something to overexpress the genes in the heterogametic gender. So you overexpress genes on the X chromosome. That happens in fruit flies. Or you might underexpress genes on one of the X chromosomes. And that's what happens in the roundworm C. elegans. And in mammals, what happens is all but one of the X chromosomes is going to be inactivated. At some point before birth, one of the X chromosomes, and I'm going to assume it's XY or XX, for XX, one of those X chromosomes is going to be inactivated. And the lineage, the cells that result from that embryonic cell will always inactivate the same X chromosome. And then in birds, well, some of the Z-linked genes might be dosage compensated, some aren't. And so in mammals, and it doesn't really matter. It doesn't matter if it's placental mammals or non-placental mammals. But one of the X chromosomes, one or the other, will be inactivated. And in the case of marsupials, in the female, whichever X chromosome you get from your father is inactivated. And in placental mammals, you randomly pick one. In fruit flies, you simply double the expression of the genes on the X chromosome in males. And in roundworms, you cut down the expression of the genes on one of the X chromosomes. And anyway, in X chromosome inactivation, it was proposed in 1961. And it was done through cytogenetic studies of karyotypes of mammals. And in the case of a human developmental timeline, we're going to go ahead and say that human embryogenesis lasts 9 months. And up until about month 4 or so, you cannot distinguish the sex in the early embryos. But at about that time, something happens. And some of the embryos develop down the male pathway, and some are going to develop down the female pathway. And at that time, if, say, for example, your sonographer knows what they're doing, they can tell you, are you carrying a son or a daughter? And anyway, this became known as the lion hypothesis. And one of the pieces of evidence is that you can look at a karyotype in an adult, from an adult cell. And in females, you see something called a bar body. Bar body. And there's the other X chromosome. And because that X chromosome that is inactivated is basically chosen by a flip of a coin, female mammals exhibit something called variegation. And for example, a calico cat. This is the coat color that's caused by, among other things, some of the genes on the X chromosome. And this gives the female calico cat this particular kind of coat color. blotches of different colors in the fur. And the male of this same breed of cat doesn't exhibit this kind of splotchy coat color. And so the females show this. And I want to say the males are all orange, but that is a distant memory, and it's not really that important anyway. The males are a solid color. Now, in mammals, all of the X chromosomes that you have, no matter how many, are initially going to be turned on and they'll be expressed. Sometime during embryonic development, and I'm going to keep it simple, you either have one X chromosome or you have two. At some point, one of those X chromosomes is going to be inactivated. And the choice is random. And as the cells go through mitosis and eventually yield the adult, somatic cell number, the same X chromosome that was inactivated that first time, it will, as the cell divides, it will uncompact itself, it will copy itself, it will go into the daughter cells. But then in the daughter cells, that same X chromosome that was inactivated in the mother cell is going to be inactivated again. And most of the genes on that chromosome cannot be expressed. There are a couple of exceptions, but usually that X chromosome is quote unquote turned off. So basically, it's always the same one that gets turned off over and over and over again. Now, if you have more than 46 chromosomes as a human, 46 chromosomes is the normal human number of chromosomes. And so if you're a male, you have 46, 1X, 1Y. If you're female, you have 46, 2X's. And you can have abnormal numbers of sex chromosomes, especially. And you can have Klinefelter syndrome, XXY. You've inactivated one of those X's. You have one bar body. You can have Turner syndrome, X, nothing. You did not inactivate an X anywhere. You have triple X syndrome, XXX. You'll have two bodies, two bar bodies, and one active X chromosome. And so somehow your cells have to count the number of X chromosomes that they have. And here is basically how it happens. On every X chromosome, there is a region of that X chromosome called the X inactivation center. ZIC. In every X inactivation center, there is a gene, X inactive specific transcript. And this gene is active on the condensed chromosome in the bar body. And this gene makes an RNA molecule that covers up the X chromosome and inactivates it. And after this RNA coats the X chromosome, proteins are attracted to it and compact to the chromosome. And so there's, as they say, there's three phases. You have to initiate. You have to pick one chromosome to inactivate. The chromosome that you pick to inactivate assuming you only have one or two. Assuming you have two. That XIST gene is expressed. Mix that RNA. That RNA covers that chromosome. Proteins get recruited to that coded chromosome and condense it. And then you maintain that condensation in that same X chromosome that you got from one of your parents every time that cell divides. And so here is the schematics of how it works. And I don't think those schematics are incredibly useful, but that's what it does. Now, that is okay. I'll go through this one. The mammalian X and Y chromosome have different features. And there is such a thing called pseudo autosomal inheritance. And they say some genes are unique to the X or the Y chromosome. And we usually don't care about the Y linked genes. There's not a whole lot of genetic information on the Y chromosome that's of any use. And so we say any gene that's on chromosome X or Y is sex linked. And those genes that are on the X are called X linked. And those that are called Y are on that are called Y linked. Unfortunately, Y linked genes are kind of boring. They go father to son, father to son, father to son, so on and so forth. And so these genes on these X or Y chromosomes are called pseudo autosomal genes. And the inheritance pattern is very similar to autosomal genes, especially as soon as you have discovered whether these genes are on the X chromosome or the Y chromosome and not on an autosome. Now, there are regions of the X chromosome and the Y chromosome that are homologous. And these regions that are homologous help the X chromosome and the Y chromosome pair during meiosis one. And that means that crossing over between the X and the Y can occur during meiosis one. Now, crossover between X and Y is unusual, but it can happen. And so you have some conserved regions in the X and the Y chromosome that mean that these will pair during prophase one. Now, the earliest experiment that showed the existence of genes on the X chromosome was done by T.H. Morgan's lab back in the 1920s and 30s. And the story is T.H. Morgan was working with Drosophila, and he was interested in finding mutants. And at this point in time, nearly 100 years ago or so, nowadays you can buy experimental lines of organisms. So you can just go order Drosophila from a supply company somewhere. T.H. Morgan had to go out in the wild and find mutants. And he found a white-eyed fly. And that particular type of fruit fly usually has red eyes. And so he found a mutation. Now, it turned out after a couple of rounds of testing, it was found that this gene that causes the white eyes was on the X chromosome. And flies use that XY system. And so fathers transmit one X chromosome, they transmit it to zygotes that become daughters. And they don't transmit it to their sons. And a male is said to be what they call hemizygous. You have half. You're hemizygous for excellent genes. And that's in comparison to homozygous or heterozygous. And so in the 1900s, T.H. Morgan and his research group found this white-eyed fly. And they did some crosses. And we crossed the white-eyed fly with a red-eyed fly, got the F1 generation. And the white-eyed fly had disappeared, which was, expected. That would mean the white allele is recessive. And so that was obeying Mendelian laws. But then they did the F2 generation, and the white-eyed phenotype reappeared. It was expected. Would again show the white eyes were recessive. But then they looked more carefully. And if you notice, only the males in that generation had white eyes. And so what they did was they did a reciprocal cross. And so you take this white-eyed male from the F2 generation and back cross it to a red-eyed female from F1. And all of a sudden, what you observe is you get white-eyed male flies and female flies. And the best way to explain that turned out that white-eyed allele was on the X chromosome, and this reciprocal cross allowed the combination of X chromosomes in females to have that inherit that mutant white-eyed gene. And I think I'm going to stop there. It's close enough. Chapter 4 turns out to be pretty long and redundant. I am going to be looking for, I found that homework that I was looking for, but now I have to decide how many points I want it to make. And I only want you to do a subset of the dihybrid crosses, because I think there's like 10 of them you can do, and you really don't have to do all 10. Once you've done a couple of them, you can do any of them. And then I'm going to look for more of this chi-square analysis and probability calculations on the Connect website. And so that better be posted, I want to say by the end of the week, but definitely by the end of the weekend. And so things will begin to populate Connect soon.